Good evening to all. Okay. Hope you are not tired for the class. Okay. Too many classes, isn't it? So today what we'll do is, okay, we'll continue with whatever we have left last time. Okay. Last time we were talking about physical parameters. Okay. Today we'll look into chemical characteristics of water. So what are the chemical parameters we should analyze for water or wastewater? That's what we are going to look into. One is biochemical oxygen demand. How many of you have heard about biochemical oxygen demand? Okay, yeah, few of you are already heard about that one. And next one is chemical oxygen demand. Third one is total organic carbon, theoretical oxygen demand. All these things will be measuring the organic content present in the water. Okay, so mostly these are used for wastewater samples. Water samples we usually we want measure. Okay, usually we go for TOC or COD. Okay, because we are not supposed to have any organic compounds in the drinking water, but these parameters are very important when we talk about wastewater. Okay, then nutrients. Okay, nutrients, what are the nutrients usually found in water or wastewater? Okay, nutrients in water or wastewater, nitrogen and phosphorus. Okay, nitrogen and phosphorus. And where is it is coming from? Okay, nutrients coming into wastewater, or water bodies. Water bodies, it is coming through the discharge of wastewater, agriculture runoff and all. We'll see that one in detail. Then other chemical parameters are toxic chemicals, heavy metals, emerging contaminants like pharmaceuticals, personal care products, etc. Whatever I have marked in red, we'll discuss in the next class. So today we'll talk about biochemical oxygen demand, chemical oxygen demand, total organic carbon, theoretical oxygen demand, and nutrients. Okay. Yeah, alkalinity and hardness we have already discussed about. So what is biochemical oxygen demand? Biochemical oxygen demand, the definition is, it is the amount of oxygen required by the microorganism for the degradation of biodegradable organic matter present in the wastewater. Each word is important. Amount of oxygen required for the microorganism for the degradation of biodegradable organic matter present in the wastewater. That means microorganisms are present along with the organic matter. They will be utilizing the oxygen present in the system and degrading the biodegradable organic matter into carbon dioxide and water. So you know the initial oxygen and you know the final oxygen. So you know how much of oxygen is consumed by the microorganism for the degradation. So that is the biochemical oxygen demand. So that is very important for us either to design the systems or to monitor the health of our rivers and all the things. Because if oxygen depletion is happening in the river, whether the biota, flora and fauna will be able to survive in the river? No. Okay, so this is very important. Okay, standard BOD, okay, we measure at 20 degrees centigrade and 5 days. Okay, that means the wastewater along with the microorganisms containing oxygen, we mix thoroughly and keep it at 20 degrees centigrade for 5 days, and after 5 days we take it and analyze for the residual oxygen, because microbial activity is slow compared to chemical reactions. So that's why we are giving more time. And 20 degrees centigrade, that is the average temperature in the world. Okay, so that's why 20 degrees centigrade is being used. Okay, and we will see later. Okay, for India, sometimes what we do is, instead of reducing the time, we do the BOD at 28 degrees centigrade and 3 days. Okay, it is not a world accepted standard, but we get almost similar results of 5-day BOD, because as the temperature increases, what will happen to the biological activity? It is increased. Okay, so you can reduce the time. So that's why in India sometimes we go for 3-day 28 degrees centigrade, but that is not an internationally accepted one, but it will be getting almost similar results. Okay, just for information I am sharing that one. It means BOD 5 is used to find out the pollution status of streams. Okay, because when some wastewater is discharged, how the oxygen depletion is happening in the river, and you know that the river reaeration will be happening. So whether the oxygen level is going beyond a permissible limit, okay, to support the aquatic organisms and all the things. The wastewater generated in the cities or the towns situated along the river discharged after proper treatment, okay, improper treatment into the street. Okay, so when this wastewater get discharged, the oxygen level will deplete and all the things. Okay, so when this organic matter is, okay, why the oxygen level is depleting? Because organic matter, okay, microorganisms are present in the river, okay, and organic matter is there, so they will be utilizing the food, so naturally the oxygen will be consumed, the level will be coming down. Okay, but you know that the river is in contact with the air, so some reaeration also will be happening. So if your microorganism utilization rate is, okay, more than the reaeration rate, then your oxygen level will be depleting. Okay, okay, so if somebody is interested, you can go and look into Streeter-Phillips equation to look into the oxygen level of river and all after discharging wastewater. Okay. Okay, so this is the reaction involved. I already told you, organic matter, okay, organic matter plus dissolved oxygen plus heterotrophic microorganism. What is heterotrophic microorganism? The microorganism which use organic matter as the carbon source. Okay, that is the heterotrophs. Okay, okay, which use organic matter as the food or energy source. Okay, it oxidize the organic matter, you will be getting carbon dioxide, water, more cells and energy. So this is the reaction happening in the BOD bottle. So okay, this is important, provide essential conditions. Okay, that means if you have to get the BOD, you have to have microorganism, okay, dissolved oxygen sufficient amount, okay, and organic matter, then nutrients, because we all need nutrients. Similarly, microorganisms also need nutrients. Okay, okay, how to find the BOD5? Okay, so what we do is we find out, okay, you have a wastewater, you take it, dilute it enough, okay, so that whatever the dissolved oxygen available in the bottle will be sufficient for the microorganism to utilize it to organize or oxidize the organic matter. So we have to dilute the wastewater if it is strong. So initial, okay, dissolved oxygen you find out, after 5 days you find out the, okay, dissolved oxygen present in the bottle. Okay, you find out the difference and multiply by the dilution factor. Okay, you will be getting the, okay, BOD. Okay, so dilution we have to do properly. Usually the initial dissolved oxygen is around 9 milligram per liter. Okay, so at 20 degrees centigrade, you all know Henry's law. Okay, so as the temperature increases, what will happen to the dissolved oxygen concentration? Dissolved oxygen concentration will increase or decrease? It will be decreasing. And if TDS, total dissolved solids, increases, what will happen to the dissolved oxygen concentration? It will be decreasing. Okay, so okay, as per the Henry's law, it may be 9.2 milligram per liter at 20 degrees centigrade, but if you are taking the wastewater, it will be having TDS and other compounds, so your DO concentration will be reducing. Usually we get around 8, 8.5, maximum 9. Okay, if temperature is high, it will be less and all the things. Okay, so I'll give you two cases. Okay, one is initial DO is 9 milligram, final DO is 5 milligram, no dilution was done, it is a pure sample, the BOD will be 4 milligram per liter, initial minus 5 and no dilution, so 4 milligram. Second case, the initial DO is 9, final DO is 0. Okay, whether the BOD can be calculated? No. Okay, why? Yeah, it should not be less than 1. Why are you putting the condition it should not be less than 1 after 5 days? Because we don't know whether the microorganisms are getting enough oxygen for the degradation. 0 means it might have reached at 2 days, 3 days or 4 days. We don't know when all the oxygen is depleted. That means sufficient oxygen was not there for the organic matter to get oxidized by the microorganism. So that's why there is a condition that the DO final should not be less than 1 milligram per liter. Otherwise, okay, you will not be getting the correct thing. Similarly, for a valid test, okay, the consumption should be greater than 2 milligram per liter. So minimum consumption should be 2 milligram and minimum leftover should be 1 milligram. Otherwise, your BOD test will not be valid. Okay, so these are important when you do the sample analysis. Okay. Okay, so okay, it invalid. What is the reason? I have given the reason. Okay, so okay, before going to the kinetic aspects, okay, if somebody, okay, has given you a sterile glucose water, Okay, because all of you have gone to the hospital and all is and we are getting, okay, that one. So that, okay, one packet is given and they are asking you to find out the BOD. Okay, what all are the things we have to do? Okay, we can just add some water and keep it in a BOD bottle and measure the BOD. Yes, no. Yeah, who gives a correct answer? I wanted to see. Okay, 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 what all are the things lacking there for the BOD experiment? Okay, one important thing is microorganism. Your sterile glucose water will be having microorganism? No. So if you put it in a bottle, nothing will happen. Okay, nobody will be degrading the organic matter. Okay, organic matter, so it will be remaining the same. That is one thing. Second one is nutrients, because if microorganism, okay, we are seeding some microorganism, but even then if the nutrients are not there, your microorganism will not be growing properly, so you will not be getting a correct value. So if you get some industrial wastewater or any of the things where enough microorganisms are not there, you have to add microorganism. Usually what we do, settle sewage, some 5 ml we add, it will be having enough microorganism, then we do the dilution and put the thing, okay, put the thing in the BOD bottle and get the, okay, BOD test done. Okay, so that is the way usually we do. Okay, so now we'll see, okay, what is the equation and how to calculate. Quickly we go through that one, the kinetic aspect. Okay, so usually we assume that the BOD or the organic matter degradation by microorganism is first order reaction. All of you know what is order of reaction, isn't it? Yes, first order reaction means the rate of change of concentration is proportional to dc by dt. If I put it proportional to concentration. Okay, so most of the biological system we assume that it is first order reaction. Okay, so the organic matter consumption depend upon the availability of organic matter. Okay, or the rate of change of organic matter concentration. So that is what, okay, okay, we assume. So okay, if L is the organic matter present in the system, dl by dt, dl by dt means rate of change of organic matter is proportional to how much is the organic matter present in the system, that concentration gradient what is available. So where L is the organic matter, T is the time. So okay, so we can put it as dl by dt equal to minus KL. Negative sign indicate because with respect to time the organic matter concentration is decreasing. So all of you, and integrating with L, okay, L0 to LT. L0 is initial organic concentration and LT. So we will be getting, okay, Ln L by L naught equal to minus KT, or LT equal to L naught into e raised to minus KT. So your exponential equation x equal to S naught into e raised to K xt or whatever the thing, it is LT is equal to L naught. So this is the, okay, organic matter left over at time t. Okay, so what is organic matter? So what is organic matter consumed during the time t? That will be equal to L naught minus LT. So that is your BOD exerted. Okay, sorry. Okay, L naught is ultimate oxygen demand. Okay, LT is the BOD remaining. So BOD exerted is equal to L naught minus LT. So it is a very simple equation. So this is what we do. Okay, YT is equal to L naught into L naught minus L naught into e raised to minus KT, because LT is L naught into e raised to minus KT. So YT equal to L naught into 1 minus e raised to minus KT. K is the, okay, biodegradation rate constant. Okay, and T is the time in days. Usually we take it. Okay, see, okay, if you plot it, you can see that this is the initial organic matter present and it is decreasing. Okay, it will be coming like that, and with respect, BOD exerted will be increasing. Okay, so that is what, okay, so that's what I have shown here. Now, okay, why BOD incubation take 5 days? Nobody asked me that question. Okay, why should we incubate for 5 days? Why not 10 days? Why not 15 days? Why not, okay, 4 days? Any answer? No. Okay, so okay, I'll show you the graph, then you'll understand. Okay, are you anybody have seen this graph? So when we look into the BOD exerted with respect to time, you will be getting this type of a curve. This type of a curve. Initially, okay, a hump, okay, this one, then you can see a hump. Okay, so be okay any organic or wastewater, you will be having organic content as well as nitrogenous compounds, that means ammonia, okay, urea, okay, organic nitrogen, etc. Okay, so what happens is initially the microorganisms, the heterotrophs are prominent in the system. Okay, they grow much faster, their yield coefficients are much higher and all, so they will be consuming the organic matter. So that's why, okay, this is happening like this. Okay, and once the organic matter get exhausted, what will happen, your autotrophic microorganism will, okay, dominate. Okay, so the ammonia oxidation and all is done by the autotrophic microorganism, they'll be converting it into nitrite and nitrate. Okay, so and 1 mole of ammonia or nitrogen needs 1.5 moles of oxygen. So that's the reason why we are seeing this hum. So mostly BOD5 we are doing during that time, most of this carbonaceous oxygen demand will be, okay, taken care. Okay, if you go further, okay, this nitrogenous demand also will be coming into picture. So total oxygen demand will be the carbonaceous oxygen demand plus, okay, nitrogenous oxygen demand. Yes, not less than equal to 1 1 1 1 1 only. Okay, it is 1. Okay, okay, I'll go back and check it. Okay, but it is 1. Okay, because we have to make sure that, okay, microorganisms are not deprived of oxygen during the entire test. That is the reason. It should, yeah. Yeah, see that one less, okay, it should be less than or equal to 7. Okay, that second condition, what I am telling is the consumption rate should be minimum 2 milligram per liter, because we are doing lot of dilution, and you know that in the BOD, any wastewater, when you take it, you will be having reduced inorganic compounds and all the things. Okay, that also will be present. So that also may be consuming some oxygen. So we don't want to, okay, over predict, okay, overestimate the oxygen. So we wanted to make sure that, okay, okay, your dilution is correct. Okay, because for example, if you have only less BOD and you are diluting, okay, okay, means larger number of times. So if you get only 0.2 milligram per liter and you multiply by 100, okay, you'll be getting 20 milligram per liter. Okay, so that can be your, okay, analysis error or this inorganic contribution and all. So to reduce the error, we are telling that minimum consumption should be 2 milligram per liter, so that any over dilution samples will be discarded. Clear? Yeah. Okay, this is fine. Okay, why 5-day BOD? Because we are interested in the carbonaceous oxygen demand. Okay, okay, so most of the carbonaceous oxygen demand will be, okay, obtained within 5 days. So that's why we are going for 5 days. Okay, but there are disadvantages of BOD test. Okay, one is time consuming, because this fast moving time, okay, nobody will wait for 5 days. So that is one disadvantage. Second one is we have to make sure that active microorganisms are needed. I talked about the sterile glucose solution and all, isn't it? Okay, and the BOD value could be in the absence of toxic compounds. So this is another important thing. Okay, many times we do all these tests to design our treatment systems and all. So you have a wastewater sample, okay, for example, some industrial wastewater sample, and I wanted to see how much is the biodegradable organic content in that one, so that first I can design a biological treatment system, then go for other chemical and physicochemical treatment systems and all the things. But when I do the BOD, what will happen? I am taking the sample and I am diluting it multiple times. So if some toxic compounds are present, what will happen? That toxic compounds are also getting diluted. Okay, so the toxicity effect on the microorganism because of the dilution will be more or less. It will be less. Okay, okay, because the toxic compound also will be getting diluted multiple times. So the toxic effect will be coming down. So the microorganism will be degrading more organic matter. So what will happen? You will be over predicting the BOD. And if you go and design with that BOD, your treatment system, what will happen? You expect that around 70% or 80% of your organic matter will be degraded by microorganism. In actual situation, it may not be happening, because actual situation, the toxic compound concentration will be very high. Okay, that will be having much greater impact on the microorganism. So these are some of the disadvantages. Okay, and another thing is BOD test does not have a stoichiometric validity, and BOD test may vary from lab to lab and about 3 to 5% due to the seeding, because we are adding some microorganism. Okay, we are not controlling the amount of microorganism. So with respect to time, you know that if you have initial concentration x naught, your x is equal to x naught into e raised to mu t or something like that. As the x naught varies, the final microbial concentration will be varying. So any system, if you have a fixed amount of work to be done, and if you have more workers, the time needed for the work to complete it will be reducing. So, and since we don't have any standard for the initial seeding microbial concentration, there will be variation, and type of microorganism we are adding, that also will be varying from lab to lab. The whatever the wastewater sample we are adding as seed, so there will be a variation. That's why 3 to 5% variation will be there from lab to lab when we do the BOD. So you got an idea about BOD test? Yes. Anyway, I will be uploading a video. If somebody is interested how to do the BOD test, okay, it will be there as the study material. Okay, you can go through the video. Any doubt in BOD? Anybody? No. Okay, clear? Okay, and when you fill the bottle and all, we have to be extremely careful. BOD bottle, okay, I have some slides. Okay, so it's a glass bottle, but make sure that, okay, when you fill the BOD bottle, there should not be any air bubble. Okay, if any air bubble is there, your entire experiment will be wrong. And when you put the cap, make sure that there is a water seal, okay, above the cap, so that even if some evaporation happens and all, no air will be entering into the BOD bottle. So these all are the precautions we have to take. Okay, everything is there in the video. If anybody is interested, you can see that video. Okay. Okay, and this one will be giving only biodegradable organic matter. Okay, so that's the reason. Okay, quick test. Okay, chemical oxygen demand. So here, instead of microorganism, we are adding some chemical which will be supplying the oxygen for the chemical oxidation of organic matter. Okay, so the time taken can be reduced. Organic matter plus the chemical we are giving is potassium dichromate. Okay, so and we give highly conducive environmental condition for oxidation. That means high temperature and low pH. Okay, high temperature and low pH. So you get carbon dioxide, water, and chromium. Okay, here K2Cr2O7. What is the chromium oxidation state? +6. Okay, +6. Okay, so it will be getting converted to Cr3 plus. Okay, so what we are doing is, after the oxidation is over, chemical oxidation is over, we measure what is the leftover potassium dichromate. So we know initial concentration, final concentration, we will be able to find out how much potassium dichromate is consumed for the oxidation of organic matter. Okay, that is the COD test. Okay. Okay, and if if ammonia is also present, the system will be converted to nitrate. Okay, it's because it is a strong oxidizing environment. It is not only organic matter, ammonia, nitrogen also will be getting oxidized. Complete oxidation, that means whatever the oxygen demand you are getting in a COD, it will be a combination of your carbonaceous and nitrogenous nitrogenous oxygen demand, and it will be oxidizing not only biodegradable organic matter, non-biodegradable organic matter also will be getting oxidized in COD. Okay. Okay, so this one I have already told. Okay, so that is the COD test. And COD test, okay, I told you concentrated sulfuric acid we do add, okay, and to catalyze the reaction we add silver sulfate in the sulfuric acid to catalyze the reaction. And sometimes we add some other chemical also, mercury chloride, okay, because any chloride is present in the system, chloride will be getting oxidized to chlorine gas. So that one will be giving lot of interference. So to reduce that one, okay, we will be adding some other thing, because now we are not adding that one because of the mercury toxicity. Okay, if your chloride concentration is more than 2000, okay, you will be getting erroneous results, because you know that it is a highly oxidizing condition. So if chloride is present, you will be getting a high COD or low COD? If chloride is present in the water, okay, and I am doing the COD experiment, you will be getting a high COD value or low COD value? High COD value, because your chloride will be getting oxidized to chlorine gas. Okay, so these are the things we have to be little careful. Okay, so another very quick, okay, test is total organic carbon. Most of you have heard about, isn't it? Total organic carbon TOC analyzer. So here what we are doing is we are, okay, using advanced oxidation process for oxidizing the organic matter. Initially we have seen biological, second one we have seen, okay, chemical oxidation. Here we are using advanced oxidation. Okay, we are using UV radiation and high temperature, and sometimes we use photocatalysis and all the things. Okay, so okay, what will happen? Your whatever the organic matter, the aromatic compounds and all will be getting oxidized to carbon dioxide and water. So water will be trapping through the water trap, and carbon dioxide, whatever is generated, will be analyzing using IR analyzer. Okay, and we will be getting that TOC. Okay, clear? Okay, it's a, okay, so anything, okay, present. For example, you have pharmaceuticals, personal care products and all the things. Many times, okay, what will happen, even the COD may not be oxidizing everything. COD test also may not be oxidizing everything. So in such cases, okay, if you wanted to see something is present in the system, okay, this TOC analysis will help you. Okay, okay, so this is the system. You have the, okay, syringe, okay, and you'll be, this is a UV reactor or photocatalytic reactor. Many suppliers will be using different system for the oxidation, but it's an advanced oxidation system. Then you have a condenser to trap the water, then CO2 detector, IR detector, and you will be getting the, okay, TOC value. Okay, and the instruments, okay, so this is the BOD bottle I was telling you, isn't it? Around 300 ml. Okay, so don't use it as a measuring equipment, okay, because it is approximately 300. So you can see that the mouth, so you have to fill up to the mouth, then put the cap. Otherwise what will happen, some oxygen, some bubbles will be there, and that will be affecting your, and this is the BOD incubator, and this is the TOC instrument directly, and this is a COD digester. You can put it in small vials, okay, around 1.5 ml sample and 2.5 ml acid and all the things, and okay, this is the, okay, heating mandel. So there you can heat it at 150 degrees centigrade. Everything will be getting digested and we can use it. And another thing is theoretical oxygen demand. This all of you know chemistry very well. So if you know the composition of the wastewater or water, okay, the chemical composition of the thing, we can directly write the oxidation equation and calculate the, okay, oxygen demand. For example, glucose, if you take it, okay, you know that it has to get converted to carbon dioxide and water. So we know that 1 mole of glucose needs 6 moles of oxygen to oxidize that one. So theoretically we can calculate. So if you know the composition of the chemical composition of the water, then you will be able to calculate it theoretically. You don't have to go for BOD, COD or THOD. Okay, and now TOC. Okay, so here that means 192 grams of oxygen is required to completely oxidize 180 grams of glucose, or 1.06 grams of oxygen is required per gram of glucose. So BOD of 1 gram per liter of glucose solution is 106, okay, yeah, 1060 milligram per liter. Okay, so theoretically we can calculate. Okay, okay, so 106 milligram. Okay, so that is the thing. So now coming to nutrients. Okay, so what all are the major nutrients we talked about? It is nitrogen and phosphorus. Okay, and okay, nitrogen is very useful for us, isn't it? Okay, then why are you bothered about nitrogen? Okay, if nitrogen is present in the water, any problem? No problem. Okay, where there is any standard for nitrogen in drinking water? And now there is a standard. Okay, okay, yeah, why? That is what I am asking, why? Okay, can you answer? The drinking water standard is, okay, if it is in the form of nitrate, 45 milligram per liter. Okay, and okay, if it is as nitrogen we are representing, okay, the standard is 10 milligram per liter as nitrate nitrogen. So Okay, many times, okay, this is one thing, okay, we have to remember when ammonia, nitrite, nitrate, because you know that organic nitrogen get converted to nitrite, okay, ammonia, nitrite, nitrate, okay. So, okay, if you want to take the mass balance and all, each molecular weight, everything will be varying, isn't it? So, usually in nitrogen present in any aqueous system, we measure, okay, represent it as organic nitrogen, ammonia, nitrogen in the form of ammonia, nitrite in the form of, okay, uh, 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 no, nitrogen, okay, or nitrogen in the form of nitrate, everything in the form of nitrogen or individual compounds. So when you see any value, you please look into whether it is as nitrite, nitrate, or it is nitrate, nitrogen, okay, because there is a lot of difference. Nitrate, what is the weight, atomic weight, yeah, molecular weight, nitrate, okay, 14 plus 48, 62, and whereas nitrogen 14, 14. So if it is nitrate, okay, okay, it will be a high value, okay, okay, and if it is in the form of nitrogen, it is, okay, 14, okay, 62 by 14, that times less, okay. So that we have to be careful, okay. So now we'll see why, yeah, you didn't answer me why nitrate is a problem. Are you heard about blue baby disease, methanoglobinemia? Yes, what is that one? Methanoglobinemia. Uh, yeah, yeah. High amount of nitrogen or nitrate? Yeah, high amount. If high amount of nitrate is present, okay, it will be affecting the health of the babies. Their body become blue color and they will be dying. Okay, so if you consume cyanide, what will happen to us? We die. Okay, and how? Okay, what will happen to our body color? Again, it will become blue. Okay, what does it mean? Okay, okay, the body will not be having any, okay, oxygen availability. Okay, see this nitrate, okay, in infants, okay, okay, what, okay, usually they'll be feeding on milk. Milk is alkaline or acidic? Milk alkaline or acidic? Milk is alkaline, okay. So in the intestines, okay, the pH of the intestines of babies are, okay, more alkaline compared to adults. So, okay, in the intestine, okay, the nitrate convert to nitrite, okay. So that bacteria is, okay, prominent, okay. So, okay, it will be getting converted and that is having very high affinity to hemoglobin compared to oxygen. So what will happen? Okay, it will be, the nitrite will be getting attached to hemoglobin. So instead of, okay, giving the oxygen, okay, the hemoglobin nitrite complex will be there. So oxygen supply to the body will be reducing. So that is the reason the body is becoming blue and, okay, the babies are dying. So then why adults it is not a problem? Okay, what is the pH of our intestine? Highly acidic. So this conversion will not be taking place, and, okay, the hence, okay, the hemoglobin nitrite complex will not be forming. So adults it is not a problem. So babies only that is a problem. So that is why, okay, there is a standard for nitrate in drinking water. But nowadays people, okay, lot of research is going on. They are talking about nitrate, then nitrous oxide, all those things, okay. Some of these things can cause carcinogenic, okay, compounds in the body and can lead to cancer and all the things. So, many studies are coming up, but, okay, the water standard is because of this methanoglobinemia or blue baby disease. Okay, the reason is clear, isn't it? Okay, mostly the nutrients present in water or wastewater are nitrogen and phosphorus. Okay, and another reason is cause of eutrophication. Eutrophication, all of you know, it is the excessive growth of algae in the water bodies. Okay, it will prevent the penetration of sunlight to the water bodies and further affect the growth of aquatic organism and deteriorate the water quality. Okay, not only prevent the, okay, penetration of oxygen. You think about the lot of algae is going on in a water body. Algae will be living forever. The cells will be decaying. So what will happen? The decayed things will be getting into the water body and that will be exerting dissolved oxygen, and the oxygen level will be coming down, and, okay, your aquatic organisms will be dying. Okay, many of you must have seen in the lakes summertime, like a fish death. Okay. Okay, what may be the reason? Yeah, lack of oxygen. Okay, many people will tell that some industries are discharging the waste and all. It may not be that one. Okay, the reason is, okay, lot of algal growth will be there, especially in tropical region, because nutrient supply is always there. So lot of algal growth is there, and algal decay will be happening, and your water level is coming down. So okay, the volume is less and more organic load, so oxygen depletion will happen, and okay, the aquatic organisms may die off. Okay, so that is another thing. So nitrogen, okay, in fresh water contains organic bound nitrogen and ammonia. So this is known as Jadal nitrogen. Organic nitrogen from plant ammonia nitrogen, okay, can be determined by digestion and distillation. Okay, Jadal nitrogen. Okay, and sources of nitrate, okay, one is domestic wastewater, second one is natural origin, third one is agricultural lands. Okay, all these things. Okay, domestic wastewater, and okay, I am talking about, okay, your domesticated animals also. Many times, okay, lot of cow sheds and all are there, and usually we won't consider their wastewater as wastewater, and it will be getting into the nearby water bodies and all, and lot of nitrogen will be there in that one also. Okay, and organic nitrogen or ammonia nitrogen present will be getting converted to nitrate because of the action of certain group of microorganism. Okay, so are you heard about nitrifiers? Okay, organic nitrogen or ammonia nitrogen. Okay, nitrifiers will get converted into nitrite and nitrate. Okay, so this is the, okay, nitrosomonas, that's the first step, converting into nitrite. Then nitrobacter will, okay, convert it into nitrate. So this is the thing. Okay, so blue baby disease I have already explained to you. So, and another thing is, by monitoring organic nitrogen, ammonia nitrogen, nitrite nitrogen, nitrate nitrogen in the stream, we can tell whether the pollution of the river is recent or past. Is it possible? Okay, you heard about environmental forensic, isn't it? Okay, when something happened, go back and check it, how it happened. So similarly, okay, you are seeing some high ammonia concentration or nitrogen concentration in the river. So I am telling, by looking into the organic nitrogen concentration, ammonia nitrogen concentration, nitrite nitrogen, nitrate nitrogen, this four measurement will tell you the pollution status. Okay, when it happened, okay, whether it is a recent pollution or a past pollution. Is it possible? Is it possible? Yes? No? Yeah, everybody is sleeping or what? You didn't understand my question. I told that organic nitrogen, ammonia nitrogen, nitrite nitrogen, nitrate nitrogen, these four components, if I measure, and okay, by looking into their concentration, I can tell when the pollution happened, whether it happened in, okay, just now, or okay, it happened past, in the past. Is it possible? Okay, I'll show you the picture, then you tell me. Okay, so now you tell me. Yes, you will be able to tell, isn't it? Because if the recent pollution, because your wastewater, it will be in the form of organic nitrogen, and okay, it will be hydrolyzing and getting converted to urea and ammonia nitrogen. Okay, you see. Okay, so this is your organic nitrogen and, okay, organic nitrogen and ammonia nitrogen. Then with respect, okay, yeah, okay, organic nitrogen. Okay, so this is organic nitrogen. Then it will get converted to ammonia nitrogen, the concentration is increasing. Then what will happen with respect to time? Ammonia will get oxidized to nitrite, then nitrate. So ammonia concentration will be decreasing like this. Your nitrite concentration, you see, it start increasing, then it will start decreasing, because nitrate will come and it will be getting converted to nitrate, and nitrate concentration will be going up like this. So if your river is having nitrate only, what does it mean? Okay, the pollution happened long back, because everything has oxidized to nitrate. But if you have more ammonia nitrogen in the river, okay, what does it mean? Okay, the pollution happened, okay, recently, or so much of pollution is happening, the river is not able to oxidize it. Okay, so by measuring these parameters, you will be able to Okay, find out what is the pollution status. Okay, and you all know what is this nitrogen cycle. Okay, it's always happen. Okay, okay, because some nitrogen will be escaping as nitrogen gas, but okay, many things will be coming back and all the things. Okay, so now phosphorus. Why phosphorus is important? Nitrogen, we know that ammonia, if you put to the river, it will be exciting the oxygen demand. Okay, but why phosphorus is important? Phosphorus is not toxic. Whether phosphorus will create any illness like blue baby disease or something like that? No. Okay, phosphorus will not be creating any problem. Only problem with phosphorus is the eutrophication, because it is a limiting phosphorus, limiting nutrient. That means very minimal concentration of phosphorus is needed for the algae and any aquatic weeds to grow. So if the concentration is below that one, it will not be growing, but that concentration is very low. So little ammonia, little phosphorus concentration is there, you will be getting very high algal blooms and all. Okay, phosphorus is also and what is the source of phosphorus? Where is it coming from? Are we okay through our urine and feces, are we discharging phosphorus? Yes, no. Are we discharging phosphorus through our urine and feces? No. Okay, how our body gets energy? We eat food. Okay, and how because we are not eating all the time, maximum 3 times a day only we are eating. So where is energy getting stored? How the energy is getting stored in our body? ATP, GTP, okay, CTP. Okay, and how are we getting the energy back? Hydrolysis of this ATP, okay, GTP and all. ATP get hydrolyzed, what will happen? Okay, you will be getting ADP and one phosphate will be coming. So our waste will be having phosphate. Okay, that is one source. Then detergents will be having phosphate. Then agriculture runoff will be having phosphate. Okay, so naturally the wastewater and all will be having lot of phosphate because of our metabolism happening. Okay, one of the phosphate is that phosphate present in water will not cause oxygen demand, or it will not deplete the oxygen from the water bodies. It can cause eutrophication even in small concentration as low as 0.02 milligram per liter. Okay, organic phosphate. Okay, I told you ATP, ADP, RNA, DNA, RNA, all the things. And orthophosphate is the most reactive phosphate. Okay, PO4. Okay, this one. Plants use this form only. Okay, and polyphosphate, it cannot be used by plants. Okay, hence it need to be converted to orthophosphate chemically. Removal mechanism. How can we remove the phosphate? Are you heard about luxury uptake? No. You all have studied about crop rotation and all, isn't it, in agriculture school? Crop rotation. After paddy cultivation, what is the next crop we do after paddy? Yeah. Okay. Pulses. Okay, why pulses we are putting? They okay, the pulse, okay, root anybody have seen the pulse node? Okay, they can fix the nitrogen, that globules, you can see small globules, that is excess fixation of nitrogen they are storing. Similarly, phosphorus, there are certain microorganisms, they can accumulate phosphorus much more than whatever is needed for their cell synthesis and all. That is known as luxury uptake. Only certain microorganism under certain condition. So that is the way biologically we need biologically we remove phosphorus, otherwise chemical precipitation. Precipitation by adding alum and ferric chloride can remove phosphate from water. Okay, so that is about this one. Okay, you have any questions about phosphorus, nitrogen or anything? Otherwise we can scan the thing and start. Yeah, this is the first question. Yeah, very answer is so obvious. Okay, everybody answered, isn't it? No, 58.
Who are the rest you are not answering? Why? 93 have joined, right?
60 answered, 93 joined. And how many are there in the class now?
I'm sure over 140 now.
140.
130
Okay.
or 140.
Yeah. Okay. Okay. So, okay, 61. Anybody wanted to answer? Any more people wanted to answer? No. Okay. So, can we discuss the answer now? Okay, because it is stopped. Okay, 62. So, answer is obvious. More organic pollution. Okay, so that's why higher BOD indicate more organic pollution, more biodegradable organic pollution. Okay, so that is the thing. Okay, because if less microbial activity is there, you will not be getting BOD. Okay, and more dissolved oxygen is there. Okay, doesn't mean higher BOD is there, and cleaner water will be having lower BOD. So this is the only answer. Okay, so next one. Okay, why is COD often preferred over BOD for routine wastewater process control? Yeah. Okay, 61. Okay, 63. Okay. Okay, we'll discuss about the answer. Okay, obviously first answer is correct, but if this question comes for GATE exam, okay, we are forced to give mark for the fourth answer also. Okay, why? COD is not affected by toxicity like BOD. Okay, because okay, toxicity, if toxic compounds are there, COD value will not be getting affected. So if this question comes in GATE, anybody can challenge. Okay, answer expected is the first one, but the fourth answer is somebody gives also. Okay, that is also correct because it is not affected by toxicity because it is a chemical reaction. If toxic compound or not is there or not, the chemical reaction will not be identified. Okay, so clear. And the other two are COD measures only biodegradable, that is not correct. COD always equal to theoretical oxygen demand. Okay, many times it will be true. Okay, but if you have aromatic compounds and all, complete oxidation may not be happening. Okay, so that is the reason. So okay, this one and the second and third is not correct. Fourth, people can argue out. Okay, that the answer is correct. Okay, we'll go to the next one because we have only 1 more minute. Okay, why is phosphorus a key nutrient in water quality management? Yeah. Okay, so so obviously third answer is the correct answer because all other things are okay. By looking at that one itself, okay, we can make out that it is wrong. The last question, excess nutrient in water mainly cause. Okay. Yeah. Okay. Anyway, everybody will make it correct. Okay. Still, one or two people made wrong. Okay. Yeah, eutrophication. So that's what the major problem with okay excess. Okay. So that is all. We'll see next class. Okay. Thank you so much.
Thank you, Professor.